A little break my vibration numbers, so I do have to find a safe place for this room. Like, I don't have a walk one that I don't. Okay, that's not a good. Anyway, so today we're going to talk about fundamental units of light. So in this section, and again, if you look at that online textbook that I have put the link for in your syllabus and on the site, it pretty much follows the same trend, right? I did update some of that stuff, and it is all exactly there. So it is still chapter 1 in there as well. If you look at the schedule within the syllabus, it tells you which chapters in workshop you are working on each week. So it does tell you exactly what chapters to look at for that particular purpose, because some of the chapters are different. Okay. Okay, so for this, we are going to talk about diversity of life, diversity of different types of cells that we see in life. We are going to talk about different types of microscopy that we can use to examine those cells and the structures within them. And then we're going to briefly, not briefly, but we are going to discuss differences between prokaryotic and eukaryotic cells, and how we can utilize each one of them in this class or in this discipline. And finally, we'll end with model organisms that we utilize in molecular and cell biology labs, research labs. Okay? Okay. So, before I even go there, actually, so let's talk about life. What are some things that we need for living things? What is life? Oxygen. Does it have to be oxygen? Not necessarily. Not necessarily. Some organisms don't, but primarily our life do need oxygen. What else? Answer. Sure, we need water. What else? Replication. Replication. Sure. Who said that? I will know. Yeah, doesn't matter. But life in general. Okay. Yeah. Oh, okay. Yes, dear. Response. They need to be able to adapt to their environment and respond to things that happen, changes that happen. Yes, dear. They need to be able to continue to survive. So some type of energy source, availability of that is definitely needed. Okay. So let's see how many of the things we got. I think we got most of them, actually, except for one. So, living things in general, right? Life is very organized to begin with compared to inanimate objects, but they need to have some type of homeostasis, ability to maintain their internal environment so they can continue to survive and be available. They need to be able to reproduce themselves and grow and develop from things that are around them, right? So they need to utilize whatever energy source is available, whatever environment they're in, and they need to be able to utilize it to get energy so that they can continue to survive. And for that purpose, they need to be able to take the matter around them and utilize it, break it down, or do whatever they need to do in order to transform it into energy. And respond to stimuli, I did hear that from you guys as well, but they need to be able to respond to changes in their environment. So if it gets too hot, they need to be able to get away from it, right? As well as adapt to their environment as it changes over time. That's a big one, because organisms can be doing all those things, but if they cannot adapt to the changing world, they will die out, right? So they need to be able to adapt and change to the changing times. Okay, so looking at cells, all these cells, right, they vary so much in the way they look and in the way they function. But all of them are still at the basic, level doing those things. They're doing the same functions, even though they look very, very different, especially when you compare prokaryotes to eukaryotes, and even within a eukaryotic organism, there's such a large variety of cell types that are available there, each one with their own function, each one with their own cells. So here are just some examples of some of the cells that you can look at. Can you identify any one of them?
So,
Nerve cell. Yes, one of them is a nerve cell, A. And it has all these axons and dendrites to communicate with those around them. Right? What do you think is happening to the cell over here? Yeah, it's finishing division, right? Cell division. And then here is a typical, it's like a mammalian cell culture cell that you will see next in a couple of weeks in your lab as well, that you guys will be working with, a typical human or any mammalian cell culture cell. This is their cell body, and inside is their nucleus. Some of them are clusters of cells in here as well. Okay, so looking at where these cells come from, well, every single living organism that we're going to look at, they're all going to be composed of this basic unit, which is a cell. Some of them will only have a single type of cells, and many of them are going to be composed of multiple types of cells coming together to create this community that will develop that entire organism. All these cells, these are not just every time they're not coming out of nothing, right? They have to be coming from other cells only. Typically, a cell of that same type is going to divide and give you more cells of that same type. And that's true for single-celled organisms as well as multicellular organisms. Looking inside the cells, even though they are all so different phenotypically or how they look, internally, again, they all have the same structures and they all have the same basic subunits. So in there, they will have nucleotides that are going to create the information transfer systems in the form of DNA and RNA. This is an information storage system essentially, right? And this sequence information is then utilized by amino acids to create proteins, right? And these proteins are your worker bees that are going to be doing all the work that you're going to see. So the DNA and RNA, DNA is typically just an information storage molecule. RNA can have both work capabilities as well as storage transfer capabilities. But your proteins are your main working structures. They are the ones that are catalyzing most of the reactions in the body and doing all the work that you see. By the way, any questions? Just raise your hand. Questions so far? Very good. Cool. Usually like to write out like main points. I'll get through it. This one. That's everything? Mm-hmm. That's good. So how big are cells? Well, it depends on the organism, right? So you have some bacterial cells that are going to be very, very, very small, right? Your typical ribosome, which is inside the cells, right, is about 20 nanometer long, the size, so it's super tiny. However, you can have bacterial cells that are running in that size as well. But typically, these are your smaller molecules. These are only going to be visible by some really sophisticated microscopes. Nowadays, there are these super resolution fluorescence microscopes. They are usually sophisticated enough to see these. Otherwise, with our typical bright field or typical microscopes that you will see in the labs, these are not going to be visible with those. Electron microscopes are the only ones that are going to be looked at the molecular level, at the atomic level, right? Again, none of the other microscopes will be able to do that. The super resolution fluorescence microscopes typically would be able to look at larger molecules within the cell, larger structures, organelles within the cell, so mitochondria. And you see those frames of ribosomes translating, those are pretty cool to see actually. I love those. Those are visible inside a super resolution microscope. And then at a cellular level, you will have your typical confocal and the bright field and the fluorescent microscopes. The cells, most of them are not going to be visible with the unaided eye. They will require some microscopy, right? Very few cells, right, are going to be able to be seen unaided. For majority of those structures, you would need to create a slide either, or you would need to place them under a microscope to be able to see them physically and visualize them. So until we discovered microscope, typically people did not have any idea of cells, right? They knew that obviously living organisms are going to be built of something, some type of a small subunit, but they were unable to see what it was. And many of them would believe that they were different types of these structures for different organisms, not the same for everything. So the invention of the light microscope not only led to the discovery of cells, but also allowed us to see all the similarities across organisms of these cell structures and how they function within them. The light microscope reveal a lot of the cell components, basic components, right? You can see the membranes, you can see cell walls, you can see the nuclei. In some cases, you can see some other structures, vacuum and others within them. However, the fine structures of your cells could not be revealed until electron microscopy was really developed out. Other ways that we can enhance the functions of a typical light microscope are phase contrast, fluorescence, dark field, and confocal. And we're going to talk about each one of them in a second and how they function. And it's important to know what the difference is between, for example, a phase contrast versus a simple bright field microscope and confocal or simple fluorescence. So a light microscope is going to reveal the basic structure of the cell, right? That's what it's good for. In a simple light microscope, you have a condenser or a lens. You have some kind of a light source that is putting light through it. How much you open the diaphragm is going to dictate how much light goes in to your object that you're trying to amplify. The object is typically going to be on a stage. Above that are going to be your objective lenses, which are going to dictate how much you're magnifying the actual structure that is placed on the slide. Now, those objective lenses are going to, again, you can typically with the microscopes go anywhere from 40x all the way to 1,000x. What do I mean when I say that? Magnification, what about the magnification? When I say 40x, what do I mean? Yes, it's not 40 times bigger than the eyes can see. So that's something to be mindful of, that you're looking at a very wide range and you can see a lot of things. So here, for example, is a cross-section looking at using a lower magnification, right, versus something that is higher magnification. You can see a lot more details, right? And I will tell you, a difference between 400x versus 1,000x is night and day. You see a lot more resolution, a lot more structural integrity at 1,000x. So it's amazing how much more you can see within that. The other thing with the light microscope is that because it's just a single source of light coming through, especially for cells that are not stained, right? Typical cells, they're just clear. If that's the case, it's very hard to see them clearly or see anything inside them. Sometimes they are completely invisible, especially if they're small cells like yeast or bacterial. So to get them to be resolved where you can actually see any type of information on them, you have to play around with the diaphragm settings and the condenser to get a little bit of contrast. Typically in a bright field microscopy, it's better to go lower with the light intensity so you can have a little bit more contrast. between the background and what specimen you're looking at so that you can examine those cells better. They are great for cells that are stained with some substance so that you can visualize the different parts of the cell. Especially, we typically use hematoxin and eosin, so you can have your nuclei and the cytoplasm colored differentially, and you can see things a little better. Yes. Yeah, cells are dead.
Okay.
In most cases, in this case, cells are dead. When you're looking at them, they are not alive. Unless, like I could be looking at a daphnia in a little slide, and that can be alive. Or you can look at pond water sample, and they'll be alive. But in those cases, you're not staining them.
Okay.
There are light cell tracker dyes that we can use that will have lower toxicity. It's not to say there's zero toxicity, there is some, but it's lower toxicity, and you can still examine your specimens in the microscope. But they are not as good or not readily available for everything that we need to do. They can be very expensive. And in most cases, if we are going to do that, we're going to be using either fluorescence or time-lapse microscopy along with it, because we're tracking them while they're alive in an incubator over time to see what they're doing. And I'll show you some videos like that later on in the semester, where we have either cell tracker dyes in them or even without them. Using time-lapse microscopy, we are observing their movement and what they're doing and where they're. But yeah, so typically for these, the way you will use them is looking at a histology slide or cell culture that's already been stained or that you have stained yourself. Okay, so one way that we can enhance this capability of light microscopy is by using what we call phase contrast microscopy. And in phase contrast microscopy, and I'll give you something. So in phase contrast, this is the word of in phase contrast, you are adding on additional layer of formulation to the light, the way the light passes, so that you can examine the depth of structures at different points, essentially. So in phase contrast, the light path will have some areas that are going to be tethered in a way that when the light passes through, so here is your structure. Let's say that here it's passing through a nucleus, right? So it's going to end up causing a shift in the light path deeper than where there is no nucleus and it's left with just a simple structure. And so here, the light path is going to have a less shift in its movement compared to here. So because of that, you're going to see that there was a structure in there that was different at a different level. So it gives you a little bit of 3D topology and allows you to get a better contrast of the image. It's still not staying, it's still, not clear over there. So it's still a clear cell. And here then you can see your cell membrane, and then internally you can see a structure that has a different depth, right? Or that has a little bit of 3D quality. And so this allows you to get cells a little bit more resolution when they're clear and a little bit more clear, more contrast so you can visualize them better. So that's with the phase contrast. And I will show you something, another video link that we can look at it a little bit more in depth for those of you who would like to see that. And then in fluorescence, again, another enhancement of light microscopy. This is the one that we use a lot, right? Because in here, you can use fluorescent dyes to tag specific portions of your cell. So you would use antibodies, for example, against actin, right? Or you can use DAPI to dye, to put a color, fluorescent dye onto the DNA inside the nucleus. And then you can look at them very easily, right? When you do that, you see here, for example, the cell body, because it has been, the actin and the myosin filaments have been stained. And then with DAPI, the DNA content is stained, so you see a very stark difference. You can clearly see the nucleus, you can clearly see the cell body. And if the cell was dividing, for example, or doing something else, you could see exactly what layers were involved, right? So So you can imagine a lot of cool applications of something like this. I can look for my favorite protein in here. I can use an antibody against my favorite protein, fluorescently tagged, and I can see where it is. Is it inside the nucleus? Is it in the membrane? Is it in the cytoplasm? And I can trigger some kind of a reaction to it, right? By treating it, let's say, with an agent that is known to move that protein from one place to another, and then watch it go and see if my treatment worked. If my protein indeed got affected by it and shifted to the nucleus or got translocated out of the nucleus, right? For example, if we have a transcription factor, let's say P53, and we zap these cells with UV light that triggers the DNA repair pathway, and then we can watch P53 get upregulated and then get translocated into the nucleus to be a transcription factor for those genes, right? So that's a really, really cool application. And that's one way that we can get a lot of information from simple light microscopy simply by using these fluorescent dyes. Again, cells are dead, right? We've already grown them, treated them, fixed them, and then stained them. In confocal microscopy, we now combine a little bit of that phase contrast idea along with the fluorescent microscopy. In this case, it doesn't have to be, it could be just a confocal by itself. And in this case, it's going to take essentially multiple images at various parts of your specimen and then build an image so that, if you had a thick specimen, so the others, up till now, we had just a single layer of cells or a very, very thin section. So no more than a couple of layers thick. Here, we have a thick specimen, maybe a nematode that was squished and put on a slide, right? I don't know, something else like that, or a frog leg and a cross section of that. But now it's a thick specimen and we want to resolve it. Under a light microscope, it's going to be blurry. There's no way to resolve that nicely. With a confocal, you can. Because it's going to take images all across that space, it's going to give you that 3D structure and it's going to resolve it better so you can see exactly where what thing is located. What area is active, what area is not active, all that good stuff. On this page, there are also some links to some newer microscopy, really cool microscopy applications. I'm going to show you one of them. Which one do you want to slide? I asked him, but I choose for it. Sorry about that. Next time. So this is one where the applications from this particular super resolution microscopy along with a high resolution digital microscopy. This creates like a hologram of your structure that you're looking at. Both of them were combined together to look at the whole brain of the fly. And so this you can look at, let's start. And so you see the cross section on this side, on the right side, and then you see the entire brain building out on the left. So if I were to stop at any given point, right, it's going to show you the cell structures at that cross section as it's taking those images. And over here, it's building out the brain itself. And so it can do that, continue to do that to build the entire brain. So without sectioning, you can take a whole brain of a small organism and have it do these cross-section studies, right? Almost like a CT scan or an MRI, and get individual cells examined at each point. And with that, combining that with the computer programming, you can look at a section of the brain and see how the neurons were located, right? How they were existing in that space, what they were communicating with, how they were interacting with each other. So that's a super cool, again, application process. So they can even look at, if it was an active organism, you could technically even look at synapses happening inside that organism. Cool. So finally, there is this super resolution microscopy that is nowadays the new hot thing. I'm sorry. With dimensional microscopy, again, conventional fluorescence microscope, even a confocal, the resolution you get is still a little blurry, right? It's not as clean. You can't exactly measure them exactly the way they normally are. It's as if you were a little nearsighted, so it's a little bit blurred image. However, with the new super resolution microscopy applications, you can see, for example, in this case, your microtubules exactly as they are inside. And you can see a molecule moving across them or inside them. And that's, we can look at down to the 25 nanometer diameter. And we can measure it accurately using these as well. So up till now, every single thing that we were looking at, they were all using light as the mode to brighten your image and to examine the image, right? Now we move to something a little bit more sophisticated in the form of scanning electron microscopy. Electron microscopes are different in that they are going to use electron, a beam of electrons now, to transfer, to hit the image with the specimen with and create an image from that. So the scanning electron microscopy, it's throwing a beam of electron across the surface of your specimen, and it's going to use that to resolve an image out of it. It can look at obviously 3D structures in this way very, very cleanly, very, very accurately, and remake that particular cell as it is in the image. So here is an example of a human neuroblastoma cell, and over here is a pollen grain. Yes.
So like the scanning electron microscope, you're saying it's going to be a surface because they're basically reflecting electrons off the surface of the lighter.
Yes, so this is looking at the surface. It's not going internally. If you combine it with something like a CD or MRI image, you could technically do both. Yes, and there are some applications like that available. But this by itself is going to be just looking at the surface of whatever you have. So it's not doing cross sections, it's not doing any of that. It's only looking at the surface. Now this is a particular pollen grain. This is not all pollen grains, by the way. Pollen grains come in as much variety as the plants they come from themselves, and they are super, super, super. I had a student years ago while I was working in Pakistan that did a project where they were looking at antimicrobial properties of honey. From various places, and they were comparing them to Manuka honey from Australia that has the highest content of, highest antimicrobial activity. It's FDA approved for that. And we tested, so we would get specific honeys that were either from a clove, right, like clove honey or orange honey, at least on the bottom, that's what they claim. We always had to make sure that's where they came from, so we would look at the pollen grains. And the kind of pollen grains we saw, they were so vicious looking, and like, no wonder. But this is a very actually a benign looking pollen grain. Some of them can be quite aggressive. I'll bring you some pictures if you would like to see. Now, this is where something that you were talking about can be done without using additional microscopy, right? So in a transmission electron microscopy, you can see details within the cell. Now here, typically, what you do is actually you create a cryofrozen structure and you slice it open, right? So it's going to look at that cross section that you've gotten at that point. But in that specific space that you have, that little slice that you have, you can look all the way down to the ribosome. So you see your endoplasmic reticulum here with the ribosomes on it, right? Studded to it. You can see your mitochondria in these structures. So you can see amazing resolution from these type of microscopy. Yeah. Now, I will post this link in an announcement as well, by the way, before we talk about domains of life. There is a link that has really good way of showing how these work. So I'm actually gonna do it with scanning. Electron microscopy. Yeah. So you can see what I'm talking about, and then you could watch any one of them. So here you can see how it uses a beam of electron instead of a beam of light, right? And with light microscopy, you were taking the light from usually typically the bottom of the sample. Here, the beam of electron is put from top, and it hits your sample, it bounces off, and it is going to then recreate that image spot by spot where it goes to give you that entire outer structure of that particular image, right? Yeah, so it's like piecing together your structure as it goes across your specimen. So I thought it was a great visual. You know, they have cute little animations, but they're really good visually to examine the difference between each one. And again, remember this, you know, in a typical electron microscopy, you're only going to get the outer surface. And it's, if you provide it with a cross section, with the transmission, you can get that cross section very clearly labeled out. Now, we did not talk about dark field, but it is again another important way that we can sometimes examine your kind of light images, right? That are really translucent. What you can do in those cases, in a dark field, when the light comes through, it actually goes away. So the only thing that is seen by the microscope is where the light was reflected or bounced off of the surface of your cell. Anything, any light that was just transmitted goes away. It goes along the sides. Yeah, this is again just a typical one. In a dark field, you're going to see the contrast of this allows another way to get some contrast in an image that's a very translucent specimen otherwise, right? In a bright field, there will be too much light to see it. But when you create that dark background, you can see that lighted image. So it's actually that's what it is telling you, that you're essentially putting in a way that all the light that is not going through the specimen goes away, that gets scattered away, and only the part that is your specimen gets lit up. But in phase contrast, you're still getting all the light, right? But it's just scattered different. That's the only. It's the intensity difference between the more 3D structure versus the one that is flat. So yeah, so I will put in this link in the announcement when I extended like a review announcement at the end of each week. So you can access it and look through these for all the different types of contrast. Well, that's just like going on and on and on about its own thing. Oh, interesting. That's not what I wanted to show you. It was a different thing. I guess I stopped. Oh. Oh no. Where were we? Oh, I apologize. We were right here. Okay. What's time do we have? This is like 15 minutes. Let's see how far. Currently, I'm talking. I'm slower in person. One or the other. No, I know. I felt like I was actually a little fast at something. You guys got it. Okay, so next we're going to talk about the various types of cells that are available in our world. So the three main domains of life. I think you guys did a lot of that in GB2 and 3, so you should at least know that much. You have your prokaryotes that are subdivided into bacteria and archaea, and then we have our eukaryotes. In many ways, archaea are closer to the eukaryotes than bacteria itself. But there are some places where bacteria can be a little bit similar too. But genetically, when we examine it at the end of the day, a lot more similarities lie between archaea and eukaryotes. So all of them, regardless of whether they are prokaryotes or eukaryotes, there are some common structures. All cells contain a cell membrane. Typically, it's a lipid bilayer, right? That makes the boundary outside the cells. And then you have some kind of an aqueous solution inside the cell, which is their cytoplasm. And then inside, they will contain all the substances that are needed to sustain that cell. Now, I say most of the time it's lipid bilayer because some archaea actually don't have a lipid bilayer. They have a lipid monolayer. But in that structure, they have hydrophilic heads on both sides. So it looks like this. So just one long hydrocarbon chain that is connected on both sides with a hydrophilic head. So some archaea are indeed the rule breakers in there. Okay. Another difference that your archaea and bacteria have, which is why, so the biggest difference between archaea and bacteria are that archaea can survive in very hostile environments, right? That's where you typically see them. And the reason for that is also that they do not contain that peptidoglycan. Their lipids are different in the lipid bilayer, but also they don't have the typical peptidoglycan like bacteria has. They have a peptidoglycan-like structure, but it is more resistant to the outside sources, and that's what is important to know about. Okay, and then cells, based on how they are organized within the inside, can be either eukaryotic, where they have a central command center in the form of nucleus, or they can be prokaryotic, where there is no nucleus. Throughout the rest of the semester, I will keep bringing archaea up when there are similarities between them and eukaryotes to kind of highlight how it is that we find that they are closer to eukaryotes. Okay, so prokaryotes, like I said, are simpler structures. Maybe they're not simpler, but just more efficient. Who knows? They do not contain membrane-bound structures, which are also called organelles. Instead, they have all their information within that one space of the cytoplasm, right? So the name prokaryote itself essentially translates to before a nucleus or no nucleus. They come in a variety of shapes and sizes still, right? You can have rod shape, you can have plate shape, you can have round, right? So there is still a lot of variety within there, and they can live in a larger variety of environments than any eukaryote would ever do. So they are able to survive. Many of them are thermophiles, or they can live in very hostile, hot springs or any such thing. So they have a really big variety of spaces that they can survive in due to their coping methods. There are more diverse prokaryotes and more numerous prokaryotes on Earth. Those are going to be your most common organisms there, right? And they are, again, divided into their two domains, bacteria and archaea. Even archaea has diversity in shapes that you see, not as much as bacteria, but they still have diversity within them as well. You typically see archaea that are either rod-shaped or plain. So the prokaryotes, here are some examples of prokaryotes. These are sulfur bacteria. They get their energy from hydrogen sulfide. So they can survive in very, very crazy situations, right? Some bacteria actually contain chlorophyll-like structures and chlorophyll that can allow them to be photosynthetic. So that's super cool for them as well. So they have another way to make their own energy, and they can survive. And then if you look at them internally, they're going to have typically their membranes. Sometimes they have that cell wall, right? They have the cell wall, but sometimes it's thicker and more exposed than others. And then inside, they are going to be just an open space with all the stuff that is needed. Typically, the DNA is stored somewhere in there, all clustered up, and that area is called the nucleoid region, even though it's not a properly membrane enclosed nucleus. Okay. The prokaryotes themselves, the eubacteria that we typically come in contact with, they live in normal environments, typical environments that we are walking in and out of as well. So that's called soil, water, humans, other organisms, right? Archaea live in hostile environments. So they live in acid, salt, extreme cold, extreme heat. And here you can see some of that structural change that I talked about, that their fatty acid chains are connected slightly differently. They are connected with an ester versus an ether. So archaea have an ether bond, and the other bacteria have an ester bond connecting them to their hydrophilic head. And like I said, some archaeas will actually have two hydrophilic heads with a single set of chains between them. So they do not need a bilayer, quote unquote. So that's a big difference between them as well. Okay. Now, eukaryotic cells, on the other hand, are a lot more elaborate. You can have a lot of different structures within them. So they don't just house a big open space with some ribosomes and whatever, right? They have more complex structures, each one with its own little function. So they have these subcellular structures that are all surrounded by that single bilayer outside. The biggest structures that you want to be aware of are the endoplasmic reticulum, right? The Golgi apparatus for transferring things from one place to another. The lysosome, endosome, and peroxisome, which are very important in waste removal and toxin removal and digestion as well. And then mitochondria for energy production, chloroplast, again, for energy production and for taking in the light to create energy as well. So in a typical, cell, you think about cytoplasm as just this like gel that's holding everything together, but it's not just that. It's actually very, very enigmatic, and it's doing a lot of things itself, right? There are lots of moving parts within it. And then obviously all these structures are tethered in that space as well, and they're each doing their own part. We're probably going to stop in one or two more slides so that we can continue next time. So we'll talk just a little bit about the nucleus. The nucleus is, again, it's our command center. It's where all our information is stored. That's going to make us who we are, tell us what to do, when to do it. And so this particular nucleus, by the way, if you look at that, it doesn't just have a single lipid bilayer, but two, right? So you can think about, and we'll talk about that, how that might have happened, because this is a very secure building, right, within the cell, for the right reasons, right? You do not want your critical information getting out or getting destroyed or damaged. But this nuclear envelope houses, protects that nucleus and the DNA that's contained within itself. And here you can see that this is your nuclear envelope, and this is your nuclear space. And in there you will have nucleoli, and then you will have the DNA content as well. So DNA inside this nucleus, and we'll stop there, is all contained within the nucleus. In a typical nucleus, you don't see chromosomes, right? It's all kind of condensed up, decondensed in there, and just hanging on to the nuclear envelope. It's anchored to various places along it. But when cell cycle starts to happen, these chromosomes will condense and be visible in their right way. And you can actually examine all the chromosomes hanging out there, getting ready for cell division, lining up at metaphase plate. Okay, any questions? Yes, no, maybe. By the way, all these images that I'm showing you, a lot of them are going to be electron micrograph images. So you can think about the type of resolution electron micrographs can provide you, right, with that capability. I love the mitochondria, how intricate it can give you that image from two levels. So we are stopping at number 25. We'll continue from mitochondria next time. Please make sure to give me your little sheets. Yeah, if you have a sticker, you want to place it on your sheet before you give me the sheet.